This project is regarding a platform for rapid CNS infection and drug monitoring. My name is Massimo De Vittorio and I'm a professor at DTU. We target meningitis, which is one of the most time-critical infections in medicine, affecting millions of people per year globally with 100,000 of deaths. And when we have a bacterial meningitis, 1 in 6 people actually die. It is extremely important for this infection to immediately identify the pathogen in order to provide the right therapy. And if this is not done, then it is unfortunately very likely to have antibiotic resistance and treatment failure. And unfortunately, the current diagnostic is very slow. It provides results in days, which is too much. In this respect, it is extremely important to be able to analyze the cerebrospinal fluid for two main reasons. First of all, because it is possible through CSF to identify through small volume of samples the pathogen to take a rapid decision about the therapy, and therefore also for the monitoring of the antibiotic delivery and release into the brain, which is not possible to be done through the analysis of the blood because of the blood-brain barrier. So there is clearly an unmet clinical need. There's a system that simultaneously can do both the analysis of the CSF for pathogen detection and for monitoring the antibiotic exposure does not exist. In our lab, we are exploiting the combination of spontaneous Raman spectroscopy and metallic nanostructures to enable surface-enhanced Raman spectroscopy, which is a technique which provides molecular composition of fluids and material with very high sensitivity, high speed, low sample volumes in a label-free manner. And we are developing a system that is a Raman spectrometer-based reader in which a cartridge combines a microfluidic platform and a SERS substrate. And this SERS substrate does through centrifugal operation the sample pretreatment and also by embedding a SERS substrate also the detection of the pathogen and also for the drug quantification. In only 30 minutes, it is possible to do the sampling, to load the sample into the cartridge, to make through the system sample pretreatment and detection, and to have results about the pathogen and antibiotic quantification in a very fast way. The team is also composed by Goar and Yaman, who are experts in diagnostics, sensing, and drug delivery, and also with a clinical expert who is expert in CNS infection, who has access to porcine and patient samples. And we are seeking for collaborations for industrialization of the system, for the mass production of the cartridge and of the SERS substrate, and also for AI spectroscopic data treatment.